Cool. I don't know how much they can see me. So the work I'm presenting today is based on a paper that's not in preprint yet, but this is the working title, Topographic Variability of Soil Carbon and Nitrogen Stocks in Hilly Permafrost Terrain of Northern Arctic Foothills. And this work is, the collaborators on this work are my advisor, Nic Jelinski, Rose Matamala, Julie Jastrow at the Argonne National Lab with the Department of Energy, who is also the funder of my research project here, and then also in collaboration with Chenlu Ping, who's emeritus faculty at the University of Alaska Fairbanks. My preferred title is Carbon Stock Conundrums with a Side of Onions. So hopefully that'll make sense as we go through this presentation. Click. All right, so just a few slides to set the stage on global carbon stocks and carbon stocks in the circumpolar north. So on the left here, we have this beautiful graphic that I love referencing, and it shows sort of above ground and below ground carbon across the latitude of the Earth. And as we can see, in the northern regions, we have a mass amount of carbon stored below ground in the soils. And then this work with Mishra et al. From 2013 talks about model over and under predictions in the circumpolar north. Our area is kind of really focused in Alaska, which is on the northern side of this map, in the little blue area sort of on the south of Alaska. So some of the work that we're doing is working to help constrain the variability. And in Alaska, on the left, we have sort of a map of permafrost continuity zones across the Arctic north. And then on the right, we have hilly soil permafrost terrain, this long sort of title for this landscape. And this Shalef et al. Paper in 2017 showed essentially the variability within the green areas in Alaska were greater than 200% of the statewide estimates of soil organic carbon stocks in Alaska. So that's huge, right? And Alaska is a massive land area. There's tons of carbon there. That's of great concern. So we've been doing work to get a handle on the variability that's there through topo sequences that were conducted from 2014, 16, and 18. So this was done previously to my PhD. So I just sort of been working on the data since I've been here. But looking at the two sites, we have Sagwan Hills in the north area and then Happy Valley. And just to draw your attention to this blue line that sort of bisects the green map, that's the glacial extent of the last Pleistocene maximum. So setting the stage, we have glacial till, and then as that was receding, we have LAAS blowing off the outwash plains, sort of capping the glacial till sites that we're studying. And then on the right, some diagrams of the two transects that we had run. So we're not going to go deep into the sampling methodology there because there's a lot of sort of nitpicky things, and sometimes the landscape in Alaska prohibits you from being able to implement a perfect sampling design. But the basic fundamentals is that in some cases they dug soil pits and described the soil pits. In some cases they took cores, and then in some cases they sampled a pit and then took cores beneath the soil pit. So when thinking about soils, I'm very interested in education. A lot of people don't think about soils. So my go-to to first talking about soils is that I say soils are like onions, right? And so why are soils like onions? Is because soils have layers, right? And then so we can see here sort of a nice, simple, vertically stratified soil. And if you take a look at the data table that's on the right, which is mostly for illustrated purposes, it's actually really nicely set up to do work. We have one row for one observation, right, of the soil horizon, and then each subsequent row is a nice sequence of the depth measurements of the profile, right? And then what we're interested mainly here today to talk about is carbon stocks, right? So to calculate carbon stocks, you take samples from the horizons, you determine the percent soil organic carbon by loss on ignition, bulk density, you multiply all that by the horizon thickness, you do that, calculate that for each horizon, and sum it up to 1 meter, right? And that's a nice simple process for calculating carbon stocks. So back to this question of like, well, if soils are like onions, how does this cryoturbated gelosol fit into this concept of layers, right? Well, it really doesn't. And it starts to break my Shrek metaphor, which I was very sad about. Then I realized gelosols are like parfaits, right? It's just a bunch of stuff that's mushed in there, and it's not nice, clean horizonations, right? So thinking about that, again, looking at the way like the data is organized, we have a problem. You can see it in red on the depth on the depth values for the horizon, right? So 0 to 5, 0 to 13, 8 to 33, and 18 to 33. That's not so clean anymore, right? And if you were to just take a core, one single core at a location, you're missing a lot of variability that's there, right? And the data itself is simply untidy. And this is frustrating, right? And every single day I wake up, I have to deal with this. Every single day, right? Line by line. And so we've been working on a way to harmonize sort of roster soil profile data with like your normal point data. So in this process, when they did the field sampling, Nic and Chen-Lu took field scale drawings of the horizonation, and then they sampled by horizon, did the laboratory analysis on these horizons, and then my job was to digitize the profiles in QGIS, take the data, apply it to the attribute tables, and then the real work was getting into R, extrapolating the data from spatial data, and like doing a bunch of work in there that is way too much for this presentation to talk about the methods there. But in that, we're able to get to a place where we can start to estimate carbon stocks. And so here are the results that we found so far for carbon stocks in our 2 study areas. So just to sort of set the stage for this graph, I think it's probably a little bit hard to read down here. On the top, we have the site, Happy Valley in the dark color and Sagwan Hills in the lighter gray color, positioned by summit to basin in pairs across the way. So we have summit, shoulder, upper back slope, lower back slope, foot slope, toe slope, and basin. And on the bottom, we have the same data reorganized. So it's just showing the site itself. So you can sort of see the variation within site. And the big thing that we found here is the only 2 positions that were significantly different from each other in their carbon stocks was the toe slope at Happy Valley, which had about 65 kilograms carbon per square meter of soil, and compared to Sagwan Hills, which had 35 kilograms of carbon per square meter. And we did this with a 2-way ANOVA, 2K HSD sort of analysis, right? And so that's really interesting because it's essentially it's interesting because carbon stocks are not sensitive to hill slope position, right? And that's sort of very, very different than what we see in non-permafrost affected soils and like sort of your typical nicely layered onion soil, right? And so in permafrost, there's a lot of other things that are going on, right? There's ice in the ground itself. So ice stocks and permafrost can be confounding factors there. And then something we really didn't dig into is like, well, we won't talk about it. And then, so yeah, anyways, in the future, the work is going to be to continue working on the spatial harmonization process and see if we can apply those to other things and maybe work on like how could we integrate a database with that or something like that. And then additionally, this summer, we'll be going up back to the North Slope and doing 2 more topo sequences on, I think, on a position it's a little bit farther north and less or possibly no lust cap on top of glacial tilt for the setting there. So maybe there's differences there. Another subtle aspect was the aspect of these 2 sites in that they're both north facing. And so there might be some changes there. But again, in the circumpolar north, the wiggles are aligned different, right? Like there's more sun up there. It's slightly different. So the influence of aspect is going to be mitigated to some degree. Yeah, so that's all I got for you. So I hope you enjoy the presentation. Any questions, comments, concerns? Satish?
I have a question, Irfan. Is there any explanation of your data by referring to the differences in bulk density or soil depth for the two sites.
Okay, so sorry. So Carl was asking a question. Also, I'm going to answer Carl's question, and then I'll come back. I'll come back to here. So Carl's question was, how do these soils form? They are gelisols. So the definition of a gelisol is that it has ice or permanently frozen ground for more than 2 years, at least 100 or 200 meters, depending from the soil surface. They're formed because it was much colder in a previous environment, and there were literally like glaciers right there. And so a lot of this is something I like getting into new learning for me about the Yodema ice formation and these like the really like intense glacial history is sort of new for me. So in terms of like age of the landscape or when exactly it formed, I think I have a little bit more work to do to understand that. I don't know if it's a full answer, but something. Okay, so sorry, Dr. Mola.
All right, yeah, thanks for your presentation. It was really great. Can you explain any of the differences or non-differences in soil organic carbon based on differences in bulk density?
Yeah, so we actually we sort of explored that in a once. Well, we number one, we didn't. In this case, we aren't able to, but we did investigate. We sort of like we did this process where we classified the soils into sort of generally mineral, cryoturbated, and sort of organic. So they're organic soils. So there are some pistils that weren't cryoturbated soils. And what I noticed in the data, there are definitely clear trends in sort of how would you say it? Like by great groups, so like histels and turbels. If you dive down into that a little bit more, when you break it down by the organic matter type, there's sort of breakouts in the bulk density that I've noticed between sapric, fibric, and hemic. And so we did sort of like a rough classification of those groups, and we're interested if there are any differences there, which might get to some of the bulk density things. And we didn't find anything, and I don't know how deeply we explored that. And then additionally, another concept I'm trying to get around is like bulk density is like mineral soil, and so then a lot of this stuff is ice volume, right? And so ice volume, bulk density is on air dried soil. If you take a core that's ice and you air dry it, all the ice is gone, right? So there can be some extremely low bulk densities within the depth of the core. And I think maybe, and this work really was only focused on the 1 meter depth. We tried to core to 300 meters in many cases, and that bulk density depth relationship might express itself a little bit more, but nothing significantly different in our study data so far, I believe. And Nick nodded, so I think that was a good answer. Jelinski.
Of that, I was just asking politely that how far are we until we just take a photo of the soil and then try to carbon storage?
I'm going to graduate in 2025. So 2 years, Jelinski. That's I'll stay here. Yeah. Down to a meter. Yeah, this is all data that was for, we had a 1 meter cutoff. We have soils that like we just cored until the auger stopped or whatever. Well, right. And so that gets right, gets into this question, right? And it's something that we're starting to explore a little as a result of this work more deeply, like how much variation is a pit? How many times should you core if you're going to core in a certain area? How many times, even in a pit? And the other thing that's something to think about out here is like the relative pedon scale of what a pedon is. Your typical idea out here is like 1 meter, but in pattern ground features where this is like where a lot of the stuff that we are is like the full soil pedon unit is like 1 meter, 2 meter, or even larger across, right? So like to really capture like one conceptual unit of soil, it's even beyond 1 meter. So depending on where you sample on the edges or on the inside will influence the carbon stocks. Yeah. Yeah, okay. So the question was like, how long is the distance of how long is the hills, how long are the hill slopes, right? So here is, you got to do a little bit of math because our 0 axis starts not exactly on the top. We pulled it back so you can see the summit, but essentially, you know, 500 or so for Sagwan Hills and 800-ish for Happy Valley meters over about 10 to 20 meters elevation change.
Any more questions? Otherwise, we'll thank Irf and we'll start to wrap up.
Thank you.